Hi, class. Welcome back. It's Dr. Lindner. We are still talking about the cell, and we are on the cell membrane. Now, in order to understand how things pass through this cell wall, or what direction things may travel, how do we know if things are traveling into the cell or traveling out of the cell? One of the key principles that we have to understand is this one called a concentration gradient. Now, there's a different concentration of chemicals between one side of the plasma membrane and the other. So if this is the cell, you may have more sodium outside extracellularly and have more oxygen outside extracellularly. But intracellularly, we have lots more potassium. Okay. So a concentration gradient tells us that things tend to move from where there's a higher concentration, meaning an area where there's more of something, to an area where there's less of something. So if there's more sodium outside of a cell, it would want to move intracellular from outside to in. If there's more potassium inside the cell than out, then potassium wants to go from where there's a high concentration to a lesser concentration. Okay, that's concentration gradient. An electrical gradient is the difference in concentration of ions between one side of the plasma membrane and the other. And what's happening is that positives and negatives tend to attract. So if there's positives on the outside, it'll want to move to the inside. If there's negatives on the inside, it'll want to move to the outside. And this keeps going back and forth because opposites attract. And by doing that between the concentration gradient and the electrical gradient, it creates what's called an electrochemical gradient. Let's just take a quick 2-minute video and listen to the transport that takes place here. Let's see if we can start this. Give me a second. There we go.
Like the walls of a house or building separate the interior from the outside world, the cell membrane is a barrier that separates the contents of the cell from the external environment. Cell membranes are a mosaic of protein and lipid molecules, both of which can drift from place to place within the membrane. Most of the surface area of the membrane consists of a type of lipid called phospholipid. Each phospholipid molecule has a polar head and a nonpolar tail. The phosphate-containing head is hydrophilic, or water-loving, because its polar structure is attracted to water molecules, which are also polar. The fatty acid tail is hydrophobic, because it is nonpolar and repels water molecules. Cell membranes are phospholipid bilayers, a double-layered structure in which the hydrophilic heads of the phospholipids face outward. The middle of the membrane contains the hydrophobic tails of the phospholipids, an arrangement that shields them from contact with water molecules.
So this out here, this is extracellular, and here it's intracellular. And you can see the hydrophobic part is here, and the hydrophilic part is the red.
Whereas membrane lipids act as a general barrier around the cell, membrane proteins have specific functions. For example, they may act as enzymes, receptors, anchors, or channels. There are two major populations of membrane proteins: integral proteins, which are part of the membrane structure and usually span the width of both phospholipid layers, and peripheral proteins, which are bound to the surfaces of the membrane.
That's a good little review of just some of the different types of proteins that we have within the cell wall. Now, let's understand how things can pass through. Now, things can pass through passively. Now, when things pass through passively, what that means is that passive does not require energy. It doesn't require ATP. When it says an active process, that requires adenosine triphosphate. That takes energy. These are passive, no energy required. Active, just like when you're more active, you're going to use energy to do that. So we'll talk about simple diffusion, facilitated diffusion, and osmosis. And then we'll talk about primary and secondary transport and vesicular transport. Now, membranes can maintain a difference of concentration of substances inside versus outside of the membrane. That's called a concentration gradient. There's more oxygen and there's more sodium outside of the cell membrane, but there's more carbon dioxide and potassium inside, right? So we do that, there's going to be more sodium out here. Well, there's some sodium inside, but not as much as there is outside. And then there's more potassium inside, and there's some outside, but not as much. Okay. The membrane can maintain a difference in charge ions between the inside and the outside of the membrane, and that difference is called the membrane potential. Sometimes they'll call it the resting membrane potential. It's the difference of charge of what we have outside compared to inside. Now, substances move down their concentration gradient, and they also move towards oppositely charged areas. Positives will move towards the negatives, and negatives will move towards the positive. So based on what we see here, substances move down their concentration gradient. We're going to have sodium going from extracellular to intracellular because there's more outside than inside. And we're going to have potassium go from intracellular to extracellular because there's a higher concentration of potassium in the cell than outside of the cell. That is passive. That does not require energy. Not when things move down their concentration gradient. The only time we need energy or ATP is if things move against. If things move against their concentration gradient, that's like swimming against the tide, walking against the wind. If the wind has your back, it's easier to move. But if you're walking into the wind, it takes more energy, more ATP. So let's just take a look at some simple diffusion, right? So here is water, and they put some blue dye here, and this is in the beginning. Now, what happens? Well, intermediately, right, this is the beginning, intermediate, you start to see that the dye went from where there was a higher concentration to where there was a lesser. So it's starting to move until equilibrium is met, right? That's just the concept of simple diffusion, things going from a higher concentration to a lesser concentration. We've seen this happen when someone wears a heavy perfume or a heavy cologne, right? They walk into a room and then boom, it goes from where there was none, right? It goes from where there was lots to where there was none. Now, passive transport, facilitated diffusion. Facilitate, if I'm facilitating you, I'm helping you. That's what this basically means. I facilitated the process. So transmembrane proteins, they can help solutes that are too polar or too highly charged to move through the plasma membrane. So it's either called channel mediated, or carrier mediated, right? It's either a channel or a carrier bringing it in. So it's either channel-mediated facilitated diffusion or carrier-mediated. Maybe in that cell membrane, maybe there's a carrier protein that's a glucose carrier protein, or maybe there's a channel, and that channel is a calcium channel. That's going to facilitate the movement. So here is extracellular. Here is intracellular, or the cytosols. This is outside the cell. This is inside the cell. That's your phospholipid bilayer. We're seeing that the concentration gradient works like this, from where we have lots of stuff to where there's little. It moves from a higher concentration to a lesser concentration. Things moving down the concentration gradient. If we're going this way, we're going against the concentration gradient, because we're going from where there's less to where there's more. So simple diffusion is that things are small enough that they can passively slip right through the membrane. That's simple diffusion. Now, the facilitated, here is a channel, and here is a carrier. This happens to be glucose because it's a 6-carbon sugar. Glucose needs a glucose carrier molecule to bring it in. Maybe this is calcium, and maybe that's a calcium channel. Sodium can't go through there. Potassium can't go through there. Magnesium. There's specific ion channels for specific ions. Here is just an example of a channel-mediated diffusion. So here is potassium. We know K is potassium. And this gate is open to this channel. That's a channel. And potassium can go from inside the cell to outside. Here the gate is closed, so it's not going to allow it. So channels have gates, and they could be opened or closed. This is called a gated channel. This is a carrier mediated. Carrier mediated, like a glucose carrier molecule. Here's glucose. It's coming in. This is the glucose carrier transporter, or the glucose carrier molecule that helps to transport and move it in the cell. It can't enter without a carrier. It just cannot. The body's not set up that way. Now, osmosis, this involves water. It's the net movement of a solvent through a selectively permeable membrane from an area of high concentration to low concentration. So it's kind of like a concentration gradient, but this doesn't involve solutes. This involves water. So if we look on the left at the start of the experiment, we have this tube, and right in the center, right here, there is a selectively permeable membrane. That's the dotted line right down the center, which means that water and these solutes can move in either direction. Now, it looks like there's the same amount of water on this side than there is on this side, because if we look at the water table, it's about even. But if I were to ask you to think about this and say, where is there more water, right? Think of what happens if this is a glass and this has water, and this is a glass, and the water level is lower. It's here. But what happens if I put in ice cubes in this? If I dump in ice cubes in here, what happens to the water level? The water level will rise, giving the appearance that it's even. And that's exactly what's happening here, because these are ice cubes on this side that raise the water table here. The ice cubes are solutes. Now, water goes from where there's a higher concentration to where there's less. So there's more water on the left, And there's less water on the right because a lot of the space is being occupied by solutes. So as a result, water gets pulled in this direction. Okay, that is osmosis. Osmosis is the pulling force of water, and water goes from where there's a higher concentration to where there's a lesser concentration. Now, how does this matter? How does this play into effect? So when we think about the pulling force of water, the force that attracts water into a system, that's osmosis. There's always water being pulled into the kidney. There's always blood and water that's being pulled in there. And high protein diets can put strain on the kidneys. We've heard that before. So dehydration forces the kidneys to work harder to clear toxins from the blood. That's why it's important to stay hydrated. But high protein diets dehydrate you. They dehydrate you because the increase in protein leads to a buildup of nitrogen in the blood. Why does it build up nitrogen? Because remember, a protein is when you have, it's an amino acid. So you have nitrogen bonded this way. So as the body is breaking down proteins, the nitrogen is released in the blood and it ends up in the kidneys to form urea. And in order to clean that out, you have to urinate it out. And when you increase in urination, you dehydrate yourself, putting strain on the kidneys. And that's a problem because the kidneys are a really important organ for us. It filters the blood, it regulates blood pressure through using renin, angiotensin 1 and angiotensin 2. And the kidneys are responsible for increasing the number of red blood cells by releasing something called erythropoietin, known as EPO. EPO is erythropoietin. So if your kidneys recognize that you're hypoxic, meaning that your oxygen levels are too low, then the kidneys release erythropoietin. It goes to the bone marrow, and the bone marrow pushes out more red blood cells. The more red blood cells you have, the higher the potential of carrying oxygen to oxygenate your tissues. In relationship to blood pressure with angiotensin 1 and 2, the way this works is that our liver makes angiotensinogen. Now, angio means blood vessel, like angiogram, angioplasty. Angio means blood vessel. Tensin sounds like tension. And ogin means it's inactive. So we have something that's inactive that's related to the tension of blood vessels. When we think of tension in blood vessels, you should be thinking, well, maybe that's related to blood pressure. Blood pressure and blood tension are the same thing. So we have this something that's related to blood pressure, but it's inactive. And then our good friend, the kidneys, come along, and the kidneys produce renin. We know the kidneys are also called renal, right? If someone's in renal failure, they're in kidney failure. The first three letters of renal are rene. The first three letters of renin, rene. So renin converts angiotensinogen into angiotensin 1. And it's pretty safe to assume that if there's an angiotensin 1, there's probably going to be an angiotensin 2. But what is it that's going to convert angiotensin 1 into angiotensin 2? What does the conversion is something conveniently called angiotensin converting enzyme, or they call it ACE. ACE is called an angiotensin converting enzyme. It converts angiotensin 1 and angiotensin 2. Why do we need angiotensin 2? We need it because angiotensin 2 is going to constrict your blood vessels. When you constrict blood vessels, it means you're making them narrower. When they're narrower, you're going to increase blood pressure. What else does angiotensin 2 do? It increases a hormone called aldosterone. Well, all right. Well, what does aldosterone do? Well, aldosterone allows us to hold on to sodium. And if you hold on to sodium, you're going to retain water as well. If you're holding on to salt and you're holding on to water, you increase your blood volume. And if you increase the volume, you increase your pressure. So the kidneys are really, really important for regulating blood pressure, for detoxification, right? For filtering things out. The kidneys release erythropoietin if you're hypoxic or have too little oxygen. And the kidneys are involved in making vitamin D. And vitamin D is needed to regulate calcium and phosphorus balance. So osmosis is important. It's important to stay hydrated. And the general rule of thumb is you take your weight, and however much you weigh, for simplicity, let's say you weigh 150 pounds, you should be drinking half of that. You divide that in half, you should be drinking 75 ounces of water a day. If you weigh 200 pounds, you divide that by two, you should be drinking 100 ounces of water. So you just take your weight, divide it in half. That's how much water you should drink. Now, check this out. Should you drink distilled water or mineral water? Well, if you're drinking distilled water, what ends up happening, if this is water and it's distilled, which means it's pure H2O with no solutes, but over here is a glass of water with H2O, but it has potassium, it has some sodium, it has calcium. So you've got minerals. This is mineral water. So if this is just pure distilled water, let me make this a little bit bigger so I can show you what happens. If this is just pure H2O, pure water, and you have blood cells in that water, water goes from an area of higher concentration to lesser. In the cell, you've got solutes. Out here, you don't. So water is going to go into this red blood cell, and then this red blood cell is going to expand and get bigger like a balloon, and then it pops. Uh-oh, that's no good. So you want to make sure that when you drink water, it has minerals. You want to make sure it's drinking mineral water. The other problem with pure distilled water is that water and minerals go together. That's why in nature it's called mineral water. If there's no minerals in here, you're going to be pulling minerals out of your tissues into that water. Water is always looking for minerals, always. So if you're putting distilled water in, it's going to rob it from your bones. It's going to rob it from your tissues. It's very, very bad to drink distilled water. So here, this is exactly what I was trying to draw out. I forgot that the slide was coming up. In terms of tonicity, the tonicity of a solution relates to how the solution influences the shape of body cells. So right here, this is isotonic. Isotonic means everything is the same. Whatever is outside and whatever is inside is the same. There's the same number of water and there's the same number of solutes that's outside of the cell as there is inside, which means that things are going to move equally in and out. So the cell doesn't swell and it doesn't shrink. It just stays healthy. If we're put into a hypotonic solution, this means that this is pure H2O, that there's no minerals or solutes. Water goes from an area of higher concentration to an area of lesser. So the water goes in and this thing swells up. In this particular case, it's put in a hypertonic solution. That means that although it looks like there's lots of water, there's more solutes here. There's more sodium, there's more calcium, there's more minerals and solutes here, which means that in relationship to the cell, there's more water inside than there is outside, and water goes from an area of high concentration to less. So the cell, the red blood cell, loses the water content and it shrinks and shrivels. That's called crenation. When it swells and explodes, that's called hemolysis or hemolysis. So we always want to make sure that we're in like an isotonic state. That's a healthy state. You don't want to be dehydrated. You also don't want to have too much salt in your system. Just right. Now, so we learned about passive transport. Let's look at active transport. Active means it needs ATP to make this happen. Now, we know that if this is a cell, there's more sodium outside of the cell, and there's more potassium inside the cell. And what happens is sodium rushes in and potassium rushes out. But now that sodium rushes in, there's still more sodium outside than there is inside. How does sodium get back out? And how does the potassium get back in? In order to do that, it needs energy. We need ATP. So what we're seeing here is in order to create the balance between sodium and potassium, we have what's called a sodium potassium pump. And the sodium potassium pump doesn't work in an equal distribution, meaning, okay, if we're pumping 2 sodiums out, we'll bring 2 potassiums in. Or if we're pumping 3 potassiums back in, we'll push 3 sodiums back out. It doesn't work that way. The way it works in this exact ratio. It's going to export 3 sodiums out of the cell, right? So it's expelling 3 sodiums in exchange for 2 potassiums coming in. But in order to work this pump, it does take energy in the form of adenosine triphosphate, right? So sodium rushes in due to its concentration gradient. Potassium rushes out due to its concentration gradient, and that's done passively. No energy is needed for that. But now to restore it back, to bring the sodium that rushed in back out, and to bring the potassium that's out back in, we're now moving those things against the concentration gradient. Because we're moving it against the concentration gradient, we need ATP. What's the ratio that it works? 3 sodiums get pumped out, 2 potassiums come back in. Sounds like a good test question to me. Now, this is active transport that's primary. The active transport that's secondary, what this means is that as we have sodium rushing in, look what can happen. In passive transport, as sodium rushes in, something can slip out the same door. Calcium can rush out. As sodium rushes in, hydrogen can pass out through here. Okay? When things are moving passively, following like that concentration gradient, that is called anti-Porter, right? Anti, things are going against in opposite directions. So sodium rushes in, calcium rushes out, sodium rushes in, hydrogen rushes out. Symporters, like when things are working symbiotically, symporters in harmony, as sodium rushes in, ooh, look what can sneak it with it. Glucose. So this is why Pepsi and Coca-Cola and all that stuff, all the sodas are so dangerous, because they're high in sodium and they're high in sugar. So that can sneak in both. Sodium and sugar can sneak in together. That's secondary passive transport. But Look what else. This explains why people who eat steak or hamburgers, right, you may want to add salt to it. It's not just about flavor, it's about helping to assist to make sure that when proteins get broken down into amino acids, they can get into the cell, and sodium helps the amino acids drive into the cell. And that's one of the reasons why innately we want to add salt to a lot of meat and protein, not just for flavor, but to assist the amino acids to get into the cell. Pretty cool how that works. Now, going back to this concept with sodium and calcium, with this antiporters, when you know how these work, we can influence them with certain herbs and medications like digitalis. Digitalis is a drug that slows the sodium pump down. So it slows the sodium pump, which allows the sodium to accumulate inside the cell, especially the heart muscle, which means you're going to have less sodium concentration gradient across the membrane. It's keeping the sodium inside. And if we keep the sodium inside, we're going to keep the calcium inside as well. And calcium is involved in helping in the force of contraction. So they use digitalis when people have heart failure to help it beat, to help it contract. In terms of transport in vesicles, there's a term called exocytosis and transcytosis. So exocytosis is a membrane enclosed secretory vesicle, and it fuses with the plasma membrane, and it releases the contents of what's inside to the outside. And I'll show you a picture of that shortly. Transcytosis is a combination of endocytosis and exocytosis, meaning it can pull in or release contents from the cell. Okay? It can move substances from one side of the cell across it and out the other side. Okay, good place to take a break. When we come back, we'll actually get into the structures of the cell.